Here we are at Unit 7, and here is a picture of the current Martian rover. Its name is Curiosity, and here it is exploring the Gale crater at the Ray Bradbury landing site on the planet Mars, a landing site named after the famous American writer Ray Bradbury. This image is an image of Curiosity taken by Curiosity. The robot is essentially taking a selfie of itself, which apparently is what robots do. It has set up a mirror and is taking a picture of itself in the mirror. Well, this Curiosity rover, it cost billions and billions, I don't know, maybe trillions of dollars, to put together and to launch off to Mars. Why would anyone send a robot to Mars? And part of the reason is Mars is, of course, a planet very close to us in the solar system, and also it has water on it. And we know that it has water on it because it has ice caps that routinely form up in the Martian winter and then they melt away. And spectrophotometric analysis of these ice caps indicates that it's water there. And we remember one of the fundamental characteristics of living things that we saw on the very first day is cells. And remember, there are four things that cells need to have in order to be cells. One of those is they need to have a plasma membrane. They also need to have within that plasma membrane an aqueous or watery solution, a cytoplasm. And you're also going to need some kind of DNA in there. And after that, you're going to need, well, you can need some ribosomes for the DNA to communicate with, to send the information for how to make proteins to. So you need these ribosomes in there. There's some more ribosomes coming in. There's another one right there. Actually, you need a fair amount of ribosomes in order to produce all of the polypeptides and proteins that a given cell would need. So those are the four basic things: plasma membrane, cytoplasm, DNA, and ribosomes. And many organisms are basically not much more than that. There's a group of organisms known as the prokaryotes. They include things like bacteria. And they have a DNA region, they have a cytoplasm, they have ribosomes, they have a plasma membrane. They might have other stuff going on for them, cell walls, these long, thin flagella coming off of either ends, some other optional equipment, but the basic four are all there. Some organisms are like that. In fact, most organisms have this very basic, very simple cell structure. The bacteria, for instance. The bacteria are the most successful organisms on the planet. About 95% of organisms on the planet are either bacteria or organisms that are similar to bacteria. Why are they so successful? Why are they so abundant? Because they're simple and they can reproduce very, very quickly. Other types of organisms have much more complicated cells, like this kind of cell right here. This is the kind of cell that you have. It's got this big nucleus. It's got a lot of DNA. It's got the same basic four: the DNA, the plasma membrane, the cytoplasm, the ribosomes, but a bunch of other stuff, other membranous components found within. Things that have exotic names, like the endoplasmic reticulum or the Golgi apparatus or the mitochondrion. And they do a wide variety of things. They process proteins, they manufacture ATP, they remanufacture proteins, they break down other organelles, and so on and so forth. We'll look at all of these organelle functions in the next unit. Now, scientists, biologists, use taxonomy to name and classify organisms. That's what we're going to do. We're going to look at the various domains of living things, the domain bacteria, the domain archaea, and the domain eukaryota. And the major way in which we try to figure out the differences between these different kinds of organisms is by looking at their DNA, looking at the nucleotide sequences on their DNA. And this not only allows us to group them into categories, but it also tries to show us how closely related they are in terms of evolutionary history. So there are three domains of living things, three types of cells. One domain is the bacteria. The other one is the archaea, which were at one time thought to be bacteria. And then there's the eukaryota. Well, the eukaryota have the plants, the animals, the fungus, and a group of organisms known as the protistans or the protoctistans. So the first thing that we're going to do is we're going to go ahead and we're going to look at the protistans. And as mentioned before, the bacteria are the simplest forms of life. Now again, I say that they're the simplest forms. It means that they're not overly complicated. They have exactly what they need to keep on surviving, keep on reproducing. The bacteria have been here on Earth before us, and there is no reason to think that they will not be here on the Earth long after we are gone. They are the secret to their success is their simplicity. They are mostly a plasma membrane, cytoplasm, DNA, and ribosomes, just what you need. Now, again, they have other things going for them. They might have some sort of a cell wall. They might have a slimy proteinaceous capsule that keeps other organisms from attacking them. They might have these flagella that are these long sort of whip-like things that move back and forth and allow them to move from one point to another. And they might have other optional equipment, but again, they are the basic form of life. Now, the other thing about the DNA in the bacteria, also in the archaea, is that it's not held up together in what we call a nucleus. Now, of course, there's the term nucleus of the atom. This is a completely different thing. In eukaryotic cells, you have a definite nucleus. As we can see right here, that big round object in the middle, 1/4 of it has been cut away. That's the nucleus. All the DNA is contained in that area, almost all of it. And that's something that you see in our cells, in plant cells, fungal cells, and so forth. But in the bacteria, in the archaea, the DNA is, again, suspended in the cytoplasm. Another thing about the DNA is that it is there's only one strand. In our cells, we have 46 strands. Other animals, fungus, plants, they can have up to 240 strands of DNA in each cell. The bacteria and the archaea, they tend to have just one strand, and this strand is circular. Now, if you look at that illustration there, that the DNA there doesn't look particularly circular, but imagine taking a rubber band, a big old rubber band, which is circular, and now you take it and you wad it up between the palms of your hands. You would get this sort of this glob of elastic rubber that wouldn't look the least bit circular, but if you were to untangle it, pull it apart, then you would see that it was indeed circular. Same thing here. If you could take this DNA and pull it apart, stretch it out, you would find out that it is one big circular loop. As it turns out, that allows the DNA to be replicated much more quickly. Bacteria can replicate their DNA very, very quickly, and that's why they can reproduce so very quickly. So they've got DNA that is able to communicate with their ribosomes. The ribosomes, as it turns out, are a bit smaller than our ribosomes, a little bit chemically different. That will come into that'll be handy later on. But even though they have a relatively small amount of DNA, there are nevertheless millions and millions of different kinds of bacteria. And they come to us in, different sizes, perhaps in different colors. And they are found everywhere. They are ubiquitous. There are bacteria living in the clouds above, the oceans below, the soil below. There are bacteria living on your skin, living in your intestinal tract. And we need to have these bacteria on our skin and in our intestinal tract, by the way. If we didn't have them, we wouldn't be able to digest our food. We wouldn't be able to keep fungal parasites off of our skin, for instance. The bacteria are everywhere. Everywhere that a living thing can be found, bacteria will be found there as well. They are truly remarkable. So there are many, many things that we could say about the bacteria. But what we'll do is we'll look at their basic characteristics again. The cell wall there. Now, not all bacteria have a cell wall, as it turns out. They might just have the plasma membrane, but again, they might have some sort of coating around the plasma membrane that might protect them. Now, we have seen, or will see, that plants, for instance, have cell walls of cellulose. We also have seen that the fungus have chitin as part of their cell wall structure. Well, the bacteria have cell walls, but they're not made of cellulose or made out of chitin. They're made out of a particular compound known as peptidoglycan. And this peptidoglycan is, as the name kind of implies, it's part protein and it's part carbohydrate, and it's like nothing else around. Again, plants and fungus have cell walls, but they don't have peptidoglycan cell walls. Animals have no cell walls at all. They have no peptidoglycan in their structure whatsoever. So it's a structure that's kind of unique to bacteria. And as we'll see later on, we can use this difference between bacteria and other forms of life against the bacteria. Most bacteria have absolutely nothing to do with us. Those that do have anything to do with us are actually beneficial to us. You have bacteria in your gut that help you to digest your food and to do other things. But there are some bacteria that, well, they're pathogenic. They can cause disease. And we might want to try to eradicate them from our bodies. Well, one way that we can do this is to go after their peptidoglycan cell wall. We can do this. By using antibiotics that will attack the bacterial cell wall. But there's a certain problem with that, a mounting problem with that. We're running out of antibiotics, and we'll look at that a little bit later. Here's more bacteria here. You can see one in the lower right-hand corner is dividing. Here are some more bacteria. They have these what are called spirochete or corkscrew shapes to them. To me, they kind of look like seahorses. Green sausages, more corkscrews and sausages, and also little round bacteria. Those are the three basic shapes of bacteria, sort of sausage-shaped, corkscrew-shaped, or round. One of the things we can say about the bacteria is that they are unicellular. They typically have just one cell. In fact, this is basically an extremely pronounced feature of the bacteria. They are unicellular. Now, in some cases, though, they're sort of they straddle the line. Here is a whole group of bacteria. There's hundreds of bacteria here. Each bacterium is one small sphere, and if you look closely at these spheres, you'll see that they are dividing in two. Well, I mentioned that one of the secrets to the success of the bacteria is their simplicity, but the simplicity allows them to rapidly reproduce. They can divide quickly. In fact, under optimal conditions, a given bacterial cell can divide within 20 minutes, and then the two resulting bacterial cells can now divide again in 20 minutes. Now you got 4. 20 minutes later you've got 8, and then 16 and 32. And after a while you have 1,000,000, and then 20 minutes later you have 2,000,000. So that's one of their great reasons for being so successful. They can reproduce quite quickly. Also, I mentioned that they have those proteinaceous capsules around them, sort of slimy capsules around their cell walls. That helps to protect them from other organisms. Well, here's a bunch of bacteria, and they're reproducing so quickly that they are stuck to each other's protein capsule. They're reproducing so quickly they can't even separate from one another. So they now produce something that looks sort of like a multicellular organism, but we usually don't call them multicellular because they do not have different kinds of cells that do different tasks, that do different kinds of functions. It's basically just a mass of individual cells that are stuck together. So we might refer to them as being colonial. Then again, there are some bacteria that seem to be more multicellular. This is a blue-green algae here, and the blue-green algae has some cells that are a bit bigger than others, as you can see there. Here's another view of it right here. Those larger cells are involved with fixing nitrogen. They're pulling nitrogen out of the atmosphere and putting it into protein. That makes them different from the other cells, which are mostly engaged in photosynthesis. Well, that's coming kind of close to being multicellular. It's not as diverse as most multicellular organisms are, but we can say that very definitely the line is blurred. Generally speaking, though, we refer to the bacteria as being unicellular or single-celled. The cells in the chain that are not enlarged, that are not fixing nitrogen, they're engaged again in the photosynthetic process, and that makes these particular bacteria autotrophs. They are feeding themselves. Other types of bacteria are heterotrophs, and some of them are both at the same time. Another strange thing about this view of this blue-green algae is you see up over here, there's a little microbe there that has a happy face on it. What the heck is going on with that? Now, while some bacteria are autotrophs, most of them are heterotrophs. They feed on other organisms. They might feed on other bacteria. They might feed on plants, fungus, whatever. These bacteria right here, these sort of look like lemons here, are feeding on the intestinal wall of some animal or another. So they are eating other organisms. So that's the bacteria, and for a long, long time, people just thought, well, there's these single-celled organisms called bacteria, and that's all there is to it. But then something strange happened. Back in the 1970s, early 80s, people began to hear that scientists inside the Soviet Union were sequencing DNA. They were coming up with new ways to sequence DNA nucleotides, and they were finding out that there were, in fact, two different kinds of bacteria, the regular bacteria that we had always suspected were there, and a different kind that had completely different DNA. Well, the idea that Soviet biologists would have found some sort of a new life form was ridiculed immediately because the reputation of Soviet biology was extremely bad. In the Soviet Union, they had forbade the teaching of evolution because they believed that it was counter to Marxist dogma. And so consequently, they could not use evolution-based understanding of how to do things like, well, increase crop yield in important plants, wheat, corn, things of that sort. A particular Soviet biologist by the name of T.D. Lysenko believed that genetically breeding the plants to produce better strains was scientifically inappropriate and also counter to Marxist dogma. So he did things like trying to change the seeds themselves in the belief that that would somehow or another change the adult plant. Well, this adventure into Soviet-style political correctness resulted in what was referred to as the Great Agricultural Collapse of 1934 within the Soviet Union. And Soviet biology had lagged ever since. So nobody took the idea seriously that the Soviet biologists would have come up with found a new life form. Well, in the 1960s, the Soviet premier, Nikita Khrushchev, realizing that there was kind of a science gap between the Soviet Union and the West, built a series of science cities along the southern border of the Soviet Union, a couple of physics cities, chemistry cities, biology cities, and so forth. And what he did was he sent all of the best scientists, well, the ones that were left over after Stalin had them killed, sent them to these cities, and now they could talk to one another without offending their fellow citizens with their politically incorrect ideas. And, they began to flourish, and Soviet biology began to flourish, and they started studying genetics, they started studying DNA, and they started finding out how you could figure out what kind of DNA was in what kind of organism, and that's how they came up with the discovery of the archaea, a different group of unicellular organisms within the, previously thought to be within the bacteria. They found this group of organisms that had completely different DNA from the rest of the bacteria. And a lot of these organisms, these little bacteria-like organisms, these archaea, live in very extreme environments. They can be found in near freezing water. They can be found in water that's boiling. They can be found in toxic sludge and so forth. And they thought that these types of environments must have been very similar to the types of environments that you would have had when the Earth was still forming about 4 billion years ago. And therefore they figured that these organisms must be representative of the earliest forms of life, so they called them the archaea, which literally means the old ones. Now, as it turns out, they aren't any older than anything else, but the name has stuck, so they're called the archaea, this completely different group of unicellular organisms with completely different DNA. This is a picture of a thermal hot spring in Yellowstone National Park. Hot springs like this can have pHs down to, in the 1 to 3 range, and temperatures that can go from, 95 degrees Celsius and above. In 2016, a tourist to the area visited a hot spring similar to this one and decided to, lean over and test the water. and he leaned over and he slipped and he fell into the water, and his body was never recovered. The family begged the park department to retrieve his body, but it simply couldn't be done. His body dissolved in this hot acidic pool. It is extremely hostile to most forms of life, and yet you notice around the perimeter you have this sort of green, yellow, orange rainbow layer. Well, these colors are due to the existence of microorganisms, of archaea. The colors are the various photosynthetic pigments that the archaea use to gather up sunlight. These organisms happen to be autotrophs, and they live close to 95 degrees Celsius. That's pretty close to boiling at pHs of 1 to 3. They're just fine with it. The archaea are also found in toxic runoff from mines that contain things like cyanide, arsenic, and lead. They're found in alkaline hot springs. They're found in Antarctic ice caps. They oftentimes have somewhat odd appearances. They have plasma membranes with this sort of puckered appearance, and they oftentimes are grouped together in tight colonies. So they're a very different form of life. Some of them live under quite ordinary circumstances, but again, many of them live in very extreme environments where you wouldn't necessarily be looking for life, and it kind of raises the possibility that somewhere out there, someplace, there might be some sort of environment that would have living things. If the archaea can live in all of these strange environments, it stands to reason that we might find organisms in unexpected places as well. Now, again, these there they are. There's a picture of two eukaryotic cells, and you can see the nucleus in each of them, that sort of dense, round, purplish area. And the other thing that you can see in this picture is you can see these, look like extremely small grains of rice, dark blue in color. Those are bacteria. And what this shows us is the immense discrepancy in the size and the volume of eukaryotic cells versus bacterial cells. The typical bacterial cell has a diameter that is 1/100th or 1/10th the size of a eukaryotic cell. So not only are the eukaryotic cells more complex in terms of what they've got inside of them, but they're also much, much bigger. So let's have a little wrap up of the bacteria and the archaea. Together, the bacteria and the archaea are known as prokaryotes. Prokaryote, the word prokaryote means, pro means before, and karyote, it's the same word, same formation that you see in the English words kernel or core. It's referring to the nucleus. Well, the bacteria, as we just saw, they don't have a nucleus, so they're called prokaryotes. They came before the development of the nucleus, therefore prokaryotes. So one thing that we can say about them is they lack a nucleus. Also, they are typically single-celled or colonial. Now, we saw that there might be some gray area exceptions, but by and large, they're single-celled or colonial. They have no membranous organelles, those little features inside the eukaryotic cell that we saw, the mitochondria, the endoplasmic reticulum, the Golgi apparatus, and so forth. They're referred to as organelles. In the eukaryotic cell, you have a lot of membranous organelles, organelles with membranes around them. The bacteria and the archaea do not. The prokaryotes have no membranous organelles. They have some organelles. They have the ribosome, but that's it. And if they do have cell walls, and they don't always have cell walls, these are made out of that substance, peptidoglycan. They might be autotrophic or heterotrophic, or both. They could be both. I wouldn't put it past them. They might be motile or non-motile. Motile means that you can move about under your own power. Non-motile simply means that you cannot. They have a typical cell diameter. Now, this is not to say typical cell size or typical cell volume or anything of that sort. It is what it is. Typical cell diameter, 1 to 10 microns. What's a micron? Well, picture a meter, and now think about a centimeter, and now think about a millimeter, and now think of the tiniest possible measurement that you can possibly think of, and you can't even think of it. That's a micron. It's a really, really tiny measurement that's used to measure really tiny things. The typical cell diameter in the prokaryotes is 1 to 10 microns. How does that compare to eukaryotes? Eukaryotes about 10 to 100 microns. So some bacterial cells are about the same size as some very, very small eukaryotic cells, but by and large, they're about 1 to 1/100th to 1/10 the size of eukaryotic cells. And their ribosomes, the prokaryotic ribosomes, are smaller. They also have a different chemical composition. They're a little bit smaller and a little bit differently made. And their DNA, well, their DNA tends to be circular and single. And that is one of the things that helps them to reproduce so quickly. So that's the prokaryotes there, and what I would suggest at this point, maybe a little bit of an intermission. We are about halfway through this particular PowerPoint. It's a bit of a longer one than most. So you might want to take a break, do something else, and then come back, and we will get on with looking at cells. We'll look at the eukaryotic cells. Well, that was a fine intermission, and we looked prior to that intermission at the bacteria and the archaea. So those are the prokaryotic cells. What about eukaryotic cells? Well, we've looked at them a little bit here as we've looked at the prokaryotic cells. Again, eukaryotic cells much bigger. They have all of these membranous organelles within, the nucleus, the mitochondrion, the Golgi apparatus, and so forth. They are much more complex. The bacteria are sort of ecologically complex in the same way that the eukaryotes are, but the eukaryotic cells are much more structurally complex. They have more things in them and more things going on between them. And also, these eukaryotic cells can be divided up into the eukaryotes are divided up into 4 kingdoms. These kingdoms, well, the one kingdom, the protistans. This is a kingdom which includes organisms that might be mistaken for bacteria, except that they're much bigger and they have a nucleus. They're just referred to as the protistans. Then there is the kingdom Plantae, which surprisingly includes the plants. Then there is the kingdom Fungi, which includes the fungi. And then the kingdom Animalia, which, you guessed it, includes the animals. So what we'll do is we'll go ahead and we'll start by looking at the kingdom Protista. Here's a typical protistan right there. That happens to be an amoeba right there. This amoeba is in the process of engulfing a smaller organism. The amoeba can change their sort of their cell shape. They can twist into different positions, and in this particular case, the amoeba is twisting into a position to be able to engulf this what looks like a relatively very small eukaryotic organism, and it's going to dissolve it and devour it. This amoeba is able to move about. It is what we call, again, motile. Again, motile means that you can move from one spot to another under your own power, as opposed to being mobile, which means that you can be moved. This is a paramecium right here. The paramecium is a really good organism to illustrate the concept of the cilia, which we'll look at later. Those are these little hair-like things that are projecting off of it. Those sort of green lozenge-shaped things that you see around it, and the long cylindrical things, these are various algae. The paramecium can move about from one point to another by wagging the cilia, and it can now engulf other organisms, be they algae or bacteria, in that sort of central groove there. It will capture them in that groove and then push them through their plasma membrane and now take them apart from within. That means that the paramecium, like the amoeba, well, they are both heterotrophs. Now, not all protistans are heterotrophs. Here are protistans known as euglena. They're quite motile. They have that little long whip-like thing, a flagellum there. It's similar to the flagellum that we saw in the bacteria, but it's also different. They have these little greenish organelles within. These greenish organelles are called chloroplasts, and they gather up sunlight and they make sugar. So the euglena here, They're autotrophs. They're photosynthetic. The amoeba doesn't have much of a cell wall. The euglena here has sort of a softish cell wall of a sort, but other protistans, like these diatoms here, they have cell walls or cell cases that are made out of silicon, glass, in other words. They have a sort of glass cell walls. This is one called Didinium. Other protistans, like this colonial slime mold here, each cell, this is actually a mass of tens of thousands of cells, or more than that. They have cellulose cell walls like you would find in plants. And then again, the amoeba that we saw before, they have no cell walls at all. This next protistan coming up here. It's known as Vorticella. It's non-motile. It sits in one spot and waits for things to sort of fall into that opening there, that it now can take whatever falls in there and push it through the plasma membrane, and now again ingest it from within. These are heterotrophs here, and virtually all of the protistans are regarded as unicellular. There might be some arguments for some of the typical protistans to be multicellular, but most of them are unicellular. However, there are some untypical protistans, the seaweeds. The seaweeds are placed into the protista. Why would that be? Well, here's part of the explanation. Seaweeds were at one time thought of as being plants, and they were studied by plant scientists. Well, seaweeds are in no way, shape, or form plants. They do not have the same internal structure. They don't have the same life cycles. They don't have the same genetics. They are in no way, shape, or form are they plants. So the plant scientists didn't want them. So they basically kicked the seaweeds out of the plant kingdom, and they had to be stuck somewhere. So they went into the protistans. Well, the protistan people didn't necessarily want them either, but they didn't put up that much of a fuss. So the seaweeds are sort of honorary members of the protistans, despite the fact that they are massive and multicellular. So they are these sort of oddball members of the protistans, but then all members of the protista are oddball. They don't really belong anywhere. Scientists have basically just thrown them together into this kingdom called protista. So the protistans are, well, they're eukaryotic. They have that nucleus. They're single-celled. Again, you don't want to mistake them for bacteria. They are autotrophs. Some of them are photosynthetic. Others are heterotrophs. They can absorb materials through their plasma membrane or kind of engulf them and then push them through the plasma membrane. And some of them are both. Some of them are autotrophs and heterotrophs. They are typically motile. There are some few exceptions. And if they do have cell walls, they're made out of cellulose or silica. And then again, they may have no cell walls whatsoever. So the protistans are these kind of, odd little unicellular organisms that we don't normally even so much as think about. They're nowhere near as abundant as the bacteria, and remember, they're not bacteria. Even though they're single-celled, they do in fact have a nucleus. But another kingdom that's much more well known to us, microorganisms, well, that would be the plantae. This includes some of the most gigantic organisms on the planet Earth, and the most widespread and actually the most successful type of organism other than the bacteria and the archaea. The plants dominate the surface of the planet Earth. Everywhere you go, you find plants, typically coating the surface. And the reason for this is because the plants are, well, they're producers. They are the first organisms to get a good crack at that energy that's coming in from the sun. They get to it first. They're very efficient at storing that energy, and they put it into growth and reproduction, and they dominate the surface of the planet Earth. And they do this because they have that photosynthetic process. They have particular photosynthetic pigments, like chlorophyll, for instance, that are very, very good at absorbing photons. And now they have specialized intracellular apparatus to take that photon energy and put it into chemical bond energy. And they do this inside of their cells. Here's a bunch of plant cells on the surface of a plant, and you can see those little greenish objects within the cells. Well, these are organelles, and they are a particular type of organelle known as a chloroplast. And a chloroplast is essentially, it is a solar-powered sugar factory. It takes solar energy and takes carbon dioxide out of the air and water out of the ground, and puts them together to make sugar. Now you've captured the you captured the energy of the photons that are being gathered up in the chemical bonds of the sugar. Now you can do all sorts of things with the sugar. You can use it to make proteins. You can use it to make other kinds of carbohydrates. You can use it to make lipids, so on and so forth. And you can also put it into growth and reproduction. So this makes the plants extremely successful. They're found in all sorts of environments all over the earth. This is obviously some sort of tropical island, and there's not necessarily a whole lot of water here, and what water there is salt water, but these plants are able to thrive under these conditions. Also, you might notice that there's a hammock there between those two palm trees. That's my hammock, so I want you to stay out of it. Here's a desert, and in this desert, we oftentimes think of deserts. And here are these pine trees or spruces growing on this high mountain peak, extremely cold. The water is there, but it's frozen. They'll just hang on until spring when it warms up and the ice and the snow melt, and they're in business again. So they're able to survive and do well in a wide variety of different kinds of habitats. Being very Now they are photosynthetic, so they're autotrophs. Also, they are non-motile. There is no point in their life cycle when they are motile. They do not have any. Their pollen grains, which in animals we would call sperm cells, are non-motile. Their egg cells are non-motile. Their juvenile stages are non-motile. They're rooted in one place, and there you go. They're also, we might as well mention it despite the fact that it's fairly obvious, they're also quite multicellular. And because of the fact that they are, they're so well suited to so many environments, and they are able to grab up so much energy in the form of photosynthesis, this very, very elegant way of gathering up the energy needed for survival and reproduction. If we were to go to other planets one day and find something other than organisms similar to bacteria or archaea, they would probably be plants. They would, if there's starlight available, then you can go ahead and photosynthesize. It's an easy and elegant way of surviving. Now, the other thing that the plants have going for them is that cellulose cell wall there. It's a very sturdy material. It's rigid, hard, and easy to produce. And it can hold a lot of water pressure inside of cells, and also can form these sort of channels within the plant, allowing it to move water and glucose and food from one point to another. And you can take these cells that are contained within these cellulose walls. The water's under very, very high pressure, and you can stack the cells together, and they're like little bricks. They're very like little hard, rigid bricks, and now you can you can rise up into the air, and you can throw your photosynthetic pigments, which you carry in your leaves, upward and outward, and gather up more sunlight and keep on growing. So it allows them to assume many different shapes and sizes, and it also, the cellulose can compact around the outside to form bark, which now protects them from other organisms. Another thing about the plants, they have a very close relationship with human beings. Human beings have been cultivating and selecting organisms for many, for millennia, for as far as we can tell, tens of thousands of years. And one of the main organisms that they cultivate are various kinds of plants. Here we're looking at a variety of fruits and vegetables, And the strawberries there, well, the ancestor of the strawberry is a little tiny thing, smaller than a pea, but human beings have cultivated them over the eons to produce large strawberries. It's the same thing for the bananas. The original wild banana is very small. Same thing with grapes and tomatoes. But we've worked with these plants, selecting certain types of offspring, and produced a wide variety of food crops that has allowed human beings to, well, become one of the dominant mammals on the surface of the planet Earth. Also, plants, they have, they're non-motile, so they can't run away. They do have the bark that allows them to defend themselves, also, spines and prickles and things of that sort. But one of the main problems that plants have is that they can be easily eaten by animals. Well, one of the things that they will do now is they'll go to chemical defenses. They'll produce a wide variety of chemical compounds that will poison their animal predators. And who are their animal predators? Well, typically it's insects. So a wide variety of chemical compounds are produced by plants to ward off insects. Now, the chemical compounds might have one effect on insects, but a different effect on human beings. So, for instance, sage and lavender, which are prized by human beings, smell the way they do, taste the way they do, because of the chemical compounds that the plants are putting into their leaves to ward away insects. They're positively nasty as far as the insects are concerned, but human beings find them to be, well, they can have a nice aroma to them. They can have a nice taste to them. And in many cases, these chemical compounds that the plants are producing can act as medicines. In fact, about 70% of the medicines that we use are originally derived from plant sources. And in most cases, the chemical compounds that we're using are used by the plants to ward off, well, insects or bacteria or other organisms. And we are making use of this chemical defense that the plants have to produce a wide variety of medicines like belladonna, aspirin, and so forth. Environments. Now again, the plants are mostly autotrophs, but it is worth mentioning that not all plants are autotrophs. This is a plant right here called the burrowweed strangler, and this is its flowering body, but most of the plant is down under the ground, and it is sticking its roots into other plants. This plant is called the burrowweed strangler because it puts its roots into another plant called the burrowweed and pulls the glucose out of the burrowweed. So it is essentially a heterotroph. This is a plant called witch's hair, or dodder. It has no photosynthetic pigments whatsoever, no chlorophyll, and instead it simply grows into other plants and basically sucks the glucose out of the other plants. These plants are heterotrophs. Now, sometimes people will be under the mistaken impression that the Venus flytrap is also a heterotroph. Actually, it isn't. The Venus flytrap runs photosynthesis, but it does have the ability to capture flies. Those two leaves there with the pinkish color to them exude a chemical compound which sounds like rotting meat, which is, of course, pure ambrosia to the flies. And the flies will come in there, and as they're walking around trying to find this rotting meat, they'll step on these very, very sensitive cells. The cells will break, and that will cause these two leaves to collapse, and the fly is now trapped inside of the leaves. The little spines there produce kind of a cage over the fly. But the Venus flytrap does not actually eat the fly. It's simply waiting for the fly to decompose so that it can get the nitrogen out of the fly. These Venus flytraps live in soil that is very, very nitrogen poor. So they need to get nitrogen from someplace, and they do it by basically capturing flies and waiting for them to decompose. They're basically using the flies as soil. So a recap on the plants are eukaryotic, they are multicellular, they are basically autotrophs. There are some few that are heterotrophic. They are non-motile, and they have cell walls made out of cellulose, and also they have that other miracle ingredient. They have chlorophyll, and these chlorophyll molecules are locked up inside of the chloroplast organelles. So that's what the plants have going for them. The fungus, on the other hand, are a completely different matter. The fungus were at one time thought to be plants, and plant scientists were forced to study them, despite the fact that the fungus are absolutely nothing like plants whatsoever. They have one thing in common: they're non-motile. They're rooted to the ground. After that, they're nothing like plants. And we usually think of fungus as being like this right here, these sort of toadstools or these mushrooms here. Well, when you see these things, actually you're only looking at the reproductive structures of the fungus. In reality, most of the fungus is under the ground in the form of these little filaments, as you see right here, these whitish filaments that are searching down into the ground, and what they're looking for is, well, they're looking for organic matter. Fungus are heterotrophs. They have absolutely no ability whatsoever to photosynthesize. They have no chlorophyll, no chloroplast, no nothing. They release enzymes into the outside world and break down organic matter and now absorb that organic matter. So they're feeding on usually dead other organisms. And they're also quite multicellular, and they are also quite non-motile. Now, a peculiar thing about the fungus is that these filaments that they send down into the ground, they can be quite fine, quite filamentous. And they're very, very good at, well, they can pull up water just fine, but they can also pull up minerals very well. All those trace elements that we mentioned before, magnesium, molybdenum, calcium, phosphorus, and so forth, they can pull all of that stuff out of the soil very, very readily. And in so doing, and in being able to do that, they can now form relationships with plants. Here's a seedling, a pine seedling, and you can see that sort of cloudy area around the plant's roots. Well, those are the fungus. Those are the fine filamentous fungus roots, as they might be called. They are connecting, the filaments of the fungus are connecting with the plant roots. And then what's happening is the fungus is pulling these trace elements out of the soil and sending them to the plants. Now, the plants can gather up trace elements too, but they're just not as good at it as the fungus are. So the fungus are basically, in a manner of speaking, feeding the plants these trace elements, and that allows the plants to now flourish. Without the fungus, the plants don't do as well. Now, what's in it for the fungus? Well, the plant will send some glucose to the fungus so that it will be able to keep on doing this job. So they have this mutualistic relationship. They are working with one another for their mutual benefit. Now, the fungus are typically regarded as being multicellular. Sometimes they're a little bit on the unicellular side. These are yeast fungus, and they sometimes form chains of single cells, which makes them kind of multicellular, but then again, kind of not. And other times they're floating around just as a single cell. So there are a few gray areas in the definition of the fungus as being multicellular, but they basically are multicellular. And again, they live in the soil. They might work with plants. They might do all sorts of things, including parasitizing other organisms. So this is a person with a really deep-seated case of toenail fungus. You know, there's creams you can use for this, but they oftentimes take a long time to work, and it's very difficult oftentimes to get rid of a fungal infection. One, because they're actually more similar to animals than they are to plants. So if you try to give them some sort of medication, well, that would be harmful to the fungus, might also be harmful to you. So you have to pick the right medication, and also they've got that thick chitinous cell wall, and it's very difficult to get materials across that cell wall. So a lot of times it's very difficult to rid oneself of a fungal infection. The thick chitinous cell walls are in the way. So what can we say about the fungus? Well, the fungus are eukaryotic. They are multicellular. They are heterotrophs. They have an absorptive heterotrophic pattern. They are non-motile, and they have cell walls made out of chitin, and they are definitely not plants. So our last kingdom here it is right here, the kingdom Animalia. And here we are seeing a typical animal there, that being a dragonfly. One of the things that the animals have going for them more than any other kingdom that you go out is their extreme degree of motility. They can move from one place to another like no one's business. Being it, well, swinging through trees or flying through the air, swimming through the water. They are extremely motile. You get some bacteria, some archaea, some protistans that are motile. The animals do not. And that's a big deal because it's pretty difficult to go around moving about the landscape if you've got a bunch of cell walls in your body. They're too rigid. They don't allow for the type of flexibility that extreme motility requires. Now, there's another thing that the animals have going for them, and it's a big one. Here's an animal right here, moving about the landscape, being motile, but it's also doing something else. It's moving about the landscape, and it's looking for organic matter, other living things, pieces of living things, whatever it might be. But it finds this organic matter, and it sticks it right here into its pie hole. Its mouth, its prostoma, whatever you want to call it, the pie hole. It finds organic matter, sticks it into its pie hole, and now, guess what? There's a tube that goes from the mouth inside the body, forms up into a gut. So there's a gut inside. There's a tube that goes inside the body. No other type of organism has this. A tube that goes inside the body, and you can stick the organic matter inside the body, and now you can break it down by enzymes. The fungus, they release enzymes and they break down organic matter, but they have to sit there and wait for it to be broken down and absorb it through their cell walls. The animals, they don't have that kind of patience. They move about the landscape with their extreme motility. They find organic matter. They stick it into their pie hole. They now digest it within, and while they're digesting it, they're moving off to find more organic matter. Here's an animal right here. We don't necessarily immediately recognize it as an animal, but it is an animal, and I'll tell you right now, there's its pie hole right in there, and it's got a gut. In fact, this is a sea anemone. About the only thing that they have is a mouth and a gastrovascular system. That's it. Well, they've got these tentacles too, but that's the simple animal model. You move about the landscape, you've got a mouth, and you've got a gut. You gobble up organic matter and you break it down in the interior. Or you fly about the landscape, there's your pie hole, there's the gut right in there. And that's it. And in order to fly about the landscape, or to crawl, or to swim, whatever it is that you do, you need a complex nervous system to be able to analyze the environment, tell where you are in space, and where the possible organic matter might be also. Yet you have a nervous system. You have neurons. You have something akin to a brain. It might be an extremely small brain, but something akin to it. You can sense the environment in a wide variety of ways, and also that allows you to avoid being eaten by some other organism, or at least you hope so. There's another animal there. It's a rather nasty looking pie hole right there, and back yonder there's a gut. Here's another animal right here. Pie hole, gut. Another animal here. Rather pretty. Pie hole, gut. Salma hayek. Pie hole, gut. Also, the other thing that we must point out with the animals is that they are extremely multicellular. They are multicellular. Salma hayek here, extremely multicellular. Pie hole and gut somewhere down there. So that's the big deal about the animals. But here's the World Cup winning Italian football team. The Italian football team entirely comprised of animals. And what is it that makes them so great? Well, they're motile. They're highly motile, which explains the success of their midfield passing game. They're heterotrophs. Watch the meat. They have no cell walls. Can't be motile with a bunch of cell walls in the way. And they are multicellular. So amongst the eukaryotes, this is the secret to the animal success. Not that animals are all that successful. There's many other kinds of species that are not animals that are far more numerous than animal species. But that's the idea. They move about the landscape. They gobble up organic matter. They stick it into their mouth and digest it in their guts. So animals, they are eukaryotic. They are multicellular. They're heterotrophs. This time they're ingestive heterotrophs. They stick it inside their bodies. They are highly motile, and they have no cell walls whatsoever. So that's the animals. Now let's look at eukaryotic cells in general. Let's see that. In general, the eukaryotic cells have a nucleus, that we know. The prokaryotes do not have one. They have membranous organelles. They've got all sorts of little tiny bits inside the cells, which we will look at in the next unit. If cell walls are present, then these are made out of cellulose or chitin. Fungus have chitin, cellulose in the plants. We could also mention silica to be found in the protistans. Typical cell diameter here, 10 to 100 microns. 10 to 100 microns. Again, that's diameter. Don't get it down as size or volume. It's diameter, 10 to 100 microns. The ribosomes are much larger than those of the prokaryotes and chemically different. The DNA tends to be multiple and linear. Many different DNA molecules, and they tend to be in linear strips. Mentioned earlier on, the prokaryotes are able to reproduce quite quickly because they have a circular strand. I didn't really mention why that was, because it's very difficult to explain it. Just as it turns out, trying to replicate a linear strand is much more difficult than a circular strand. As it turns out, you have to start at multiple places on a linear strand, and on the circular strand, you can start at 2 places and just get the ball rolling, and you're done. So eukaryotic cells cannot multiply as rapidly as prokaryotic cells can do. So that's them. That's the eukaryotic cells there. Here's an activity for you. I believe that this is activity 13. If I've miscounted, don't worry about it. It's okay. It's just a number. It's just a name. This is activity 13 here. And we got across the top there, does the organism have a nucleus or not? Is it single or multi-celled? Is it heterotrophic or autotrophic? Motile or non-motile? Does it have cell walls? And if so, what kind? And then you got that column on the side that tells us what type of organisms we're looking at. So in that row next to the prokaryotes there, you just go across it, each column, and answer, well, does it have a nucleus or not? Yes or no. Single or multi-celled, heterotrophic or autotrophic. So, you might want to just go ahead and go over the PowerPoint and use this activity to see how much of this information you've got recorded. And your answers are going to look something like this. So that's activity 13 there. So I'll let you either stop the PowerPoint and work on that now, or come back to it later, because we're going to finish off this particular PowerPoint with one last thing. We're going to look at another group of things. They're not organisms. They don't have a domain. They don't have any kingdoms because they're not alive. They're not actually living things, but they're rather important to living things. So if you want to go ahead and roll we'll just stay here and go to the next slide here, and we will look at the viruses, our good friends, the viruses. What are viruses? Well, I'll tell you right now, they're not living. They do not meet hardly any of the criteria, any of the characteristics of living things that we saw before. Remember, to be a living thing, you have to have virtually all of those 10 characteristics. And the viruses, what do they have? Well, they're made out of organic matter. They don't have much water in them to speak of. And they do have some DNA within, but that's about it. That's the end of it. They are not really living things. They are particles, but they are particles of organic molecules, and these organic molecules can enter into our own system and, well, have consequences. Here is a picture of a virus in which it is basically looking sort of like a World War II sea mine here. And what you've got is this outer brownish coating, which is made out of protein. You have these little spikes that are coming off of the coating, and these little spikes are referred to as spikes. And inside, you've got that yellowish spaghetti there. That's a DNA strand. So what are the viruses? They're protein and DNA. That's it. There's DNA inside, there's protein on the outside. Here's another view of a different virus. Looks kind of like a 1960s telecommunications satellite, that sort of a corrugated aluminum or a checkered aluminum coating. That's the protein, known as the capsid collectively. The little iron coils inside, that's meant to represent the DNA, and then you have these spikes. What do these viruses do? And the answer is they don't really do too much of anything at all. They just float around in the environment, and those spikes, however, will allow them to bond on to the cells of organisms. They might bond onto plant cells, they might bond onto fungal cells, whatever it might be. There are bacteria that have viruses that invade them. So all sorts of organisms are susceptible to viruses. The virus makes contact with the cell of the host, the intended host, and now will shoot its DNA into the host cell. So if you are a human being, you breathe in virus particles, which we do every single day of our lives, and they make contact with our cells. If it's the right kind of virus, it will inject its DNA into our cells, and what it does next, well, it goes in there, and the DNA goes into to our cells, and uses our own cell machinery to start making copies of the DNA of the virus, the viral DNA. It turns our cells into a viral DNA factory. It also sends messages to the ribosomes, to our ribosomes, because they don't have ribosomes, and telling them to make the viral protein. So again, it's turning our cells into a virus factory. And this goes on for 12 hours, 24, 3 days, 10 days, whatever it might be. The viruses have different longevities. They run their own cycle inside our cells, and then they quit. They go away. Well, actually, that's not true. They don't go away. Their activity goes away, but actually the viral DNA remains inside your cell until that cell no longer exists. They will incorporate their DNA, the viral DNA, into our DNA, and it stays there for the rest of time. Again, they run their own cycle. Just depends upon the virus. Again, 12 hours, 24, 3, 10 days, or longer, maybe across the course of many, many years. But here's the basic idea here. It's showing us this cycle. The virus attaches to the outside of the cell, gets inside the cell. It now releases its, well, in this case, the virus in this particular instance is the protein coat on it is being dissolved. Sometimes they'll leave their protein coat outside the cell. And what do? Well, they tell our nucleus to start manufacturing more viral DNA and more viral protein, and these proteins and this DNA are now assembled inside of our cells, and they now release to the outside environment. So it's that's how the viruses work. They're not living. They just happen to have DNA that will work inside of our own cells. So they're just sort of roving packets of DNA that every once in a while connect with a living cell, and that living cell is now turned into a viral factory, a machine for making more viruses. Can't match. Here's a model of the herpes virus. Herpes is the virus that, the herpes condition. This is the virus that causes what's called cold sores, right there. And this is one of the viruses that has a much longer lifespan. It can last. Its effects can last for 20 years once the person is infected. It goes through periods of remission, and then it goes through periods of activity. But it basically beds down in nerve cells. And every once in a while, for whatever reason, basically due to some internal rhythm of the virus itself, it now begins producing more viral DNA and more viral proteins. Here's our good friend right here, SARS-CoV-2. That's the one that causes COVID-19. And it's basically it's called people call it the coronavirus, but there's all kinds of coronaviruses. It's a big family of viruses called the coronaviruses. What do coronaviruses do? Sometimes they don't do anything. For instance, with this particular virus, at least 35% of people who contact the virus have no symptoms whatsoever. Most people who do have symptoms, they have a really bad cold or maybe just a mild cold. The problem is that if people have what are called comorbidities, diabetes, obesity, heart conditions, so on and so forth, they are, for whatever reason, this virus tends to produce very dramatic effects in some people. So it's a very, very strange virus, and but you get it, you don't have any of these morbidities, it goes away. Problem is, they're beginning to think you might be able to catch it again. This is human papillomavirus. This is what causes warts. It's one of the many viruses that causes warts. This is a relatively tiny virus. This is HIV. This is the virus that causes AIDS. I'm looking at viruses that affect human beings, but the fact of the matter is most viruses do not affect human beings. They affect plants, insects, all manner of living things. Here's an illustration of a virus attacking a bacterium. You can see the peptidoglycan cell wall there, and in order to penetrate through this cell wall, it basically injects the DNA right through the peptidoglycan, and that now takes over the bacterial cell, and it becomes a viral factory, just like any other virally infected cells. So what are the viruses? The viruses are non-cellular. They are non-living. They don't meet the full criteria for life. They do have DNA, but they're not really being operated by a DNA program. They have organic molecules. They're not mostly water. They have no plasma membrane. They have no cytoplasm. They have no ribosomes. They have no cell walls. There's no cell there. They do not produce enzyme-driven pathways that convert energy and matter. They cannot even reproduce by themselves. They need other organisms, or I should say, they need organisms to do that. And they've got that protein capsule. Again, no cell wall because it's not a cell. Their typical diameter is 0.1 microns, less than 0.1 microns. This is what scientists refer to as extremely teensy, very teensy. They are particles. They're like dust floating about. And they've got DNA or RNA strand. The HIV has RNA in it. The RNA is injected into the host cell, and it is then turned into DNA by the host cell. There might also be certain enzymes inside the virus that don't engage in any enzyme-driven pathways within the virus. They're simply released into the host cell. The host cell now uses the enzyme. And there could be some other materials within this protein capsule. So viruses are non-living particles that can interact with living things. This is why antibiotics are completely useless when it comes to viruses. Antibiotics only work on living things. Principally, they work on bacteria. The antibiotics are used in most cases, to fight off bacterial infections. And most antibiotics work by targeting the peptidoglycan cell wall of the bacteria. It keeps the bacteria from being able to reproduce. This gives your immune system time to mop up the bacteria. It attacks the peptidoglycan cell wall, and therefore it cannot produce new peptidoglycan for the new cells that are to be reproduced, so they can't reproduce. Other antibiotics target the ribosomes of the bacteria. Again, their ribosomes are slightly different from ours. They have a different chemical composition. So we don't have peptidoglycan in our systems. We don't have bacterial ribosomes. So the antibiotics target the bacteria, not the human being. Now, human beings do have certain sensitivities to certain antibiotics, as they can be sensitive to any chemical compound. But the antibiotics are effective against bacteria they are not effective against viruses. Viruses don't have peptidoglycan cell walls, and they don't have ribosomes. Now also, people want to know, well, why don't we have a vaccine for this virus? Well, actually, it's pretty difficult to come up with vaccines for viruses because the viruses have these protein coats. You have to have the vaccine has to target something, and the protein coat in these viruses is difficult to target, and oftentimes it changes. The protein coat changes in the virus. This is one of the problems with HIV. The protein coat is constantly changing, so vaccines need to have something to target. It's very, very difficult to build a vaccine for a virus. Now there's talk of one for the COVID coming out. At the end of this year, next year, if that happens, they will have produced a vaccine for a virus in record speed. So there is the possibility that something like that could come about. But viruses are tricky things, and they have a wide variety of health effects on people depending upon the virus and upon the person. So these are learning objectives for Unit 7. It says be able to describe or identify differences between prokaryotic and eukaryotic organisms and viruses. And remember, viruses, not organisms, on the basis of size, structure, complexity, DNA content, diameter, and whether or not they are living. And the plants and the fungus, completely non-motile. So of all the multicellular kingdoms, the animals are the only ones that are motile. So that was Unit 7 there, and Unit 8 we're going to go ahead and we're going to look at the eukaryotic cell in much more detail. We're going to look at the various organelles, look at the names of these organelles, their structure, what they're made out of, their function, which is to say what they do. And we'll look at that in both animal cells and in plant cells. That's in Unit 8. And the reason for this is because of the fact that, there's many reasons for it, but there's one fundamental fact that needs to be considered. The animals do not have cell walls. Plants and fungus do have cell walls. To a desert like this, and there's plants growing all over the place. They love it. They have all the sunlight they can need. They get CO2. The water isn't in such great abundance, but if you dig down a little ways, you'll find some water, and that's what the plants are doing. They're putting their roots down into the soil. And also, when the water does appear, they can soak it up. They can store it up in their tissues and use it throughout the year. So this is a wonderful environment for them to grow. Bacteria, these archaea, are very, very simple. Their DNA is just sort of suspended there in the cytoplasm. It's not locked up into a nucleus as the DNA is in our cells. There's a picture of a eukaryotic cell right there. And it has a very, very definite nuclear region. The cells that we're seeing here, the bacteria and the archaea, are also quite small. Here's a microscope image of